Hello students, welcome to my channel Online Biology Tutorials. I am back again with some more questions from chapter number 11. However, MCQ is not there in your syllabus, but you should go through them because might be you can use these questions while asking any short note or a long note question. Okay, so the first question that I have picked for you is a technique associated with DNA amplification. DNA amplification is the keyword here, means they are talking about the amount of increase of DNA. So here we will quickly pick PCR, polymerase chain reaction. Production of large scale recombinant products can be done by autoclave. This is wrong because autoclave is used for sterilization. Tissue culture labs will not be the answer. Thermocycler is the another word for PCR, but here they are asking for large scale recombinant products, so answer will be bioreactor. So while if a question comes on bioreactor, then you can write this term that the production of large scale recombinant products is being done in the bioreactor, because this is a good key sentence. Next question is, which of the following is the correct sequence? Now this question may come as, what is the sequence for the process of recombinant DNA technology? So we know that this happens first, isolation of DNA, then we use to cut it, then we use to separate the fragments, then we use to ligate them, then we use to transfer them, and ultimately the host was cultured. So please do learn all these 6 steps because this is important question. What are the steps of recombinant DNA process? If you go on in the option, it would be 1, then we came on to 3 and 2. 1, 3, 2. That means this is the right answer. Restriction enzymes belong to a large class of enzymes, so they are obviously nucleases, because restriction enzymes, they act upon nucleic acids, and any enzyme that act upon the nucleic acid is known as nuclease. Which of the following is used as a selectable marker? Now selectable marker was the one that was generally antibiotic resistant gene, right? So we'll be picking up this thing. And for antibiotic, penicillin is generally not used, so it was never ever discussed to you. Don't get confused in it. And this and salmonella can never be the answer to it. What would be the molar concentration of human DNA in a human cell? So it is 0.4%. This you need to learn, and they have mentioned in chapter number 6 that it is approximately equals to 6.6 into 10 to the power 9 base pairs. Okay, so please do learn this thing. Using a single template molecule, how many DNA molecules are generated after 10 cycles of amplification in PCR? Remember, if you have 2 strands of DNA, right, which are anti-parallel, after 1 cycle they are going to yield 2, right? So the formula to calculate is 2 to the power of the cycles, right? So here we have 2 to the cycle power 2 is to 10, and if you will calculate, it will come up to 1024. This numerical question number 7 is important. You may get such type of numericals. In a vector and DNA fragments are generated using the same restriction enzyme, how can self-ligation be prevented? Until it is not suggested, they are not being ligated. So here this enzyme alkaline phosphatase is also important to learn, right? So alkaline phosphatase is used to prevent such self-ligation. Next question is, beside better aeration and mixing properties, what other advantages do stir tank bioreactors have over shake flask? So this question is given indirectly in NCERT, that bioreactors are always better over shake flasks because they provide the right pH, they provide oxygen, they provide the right temperature, they provide the right pressure, substrate, which is otherwise not possible in the overshake flask. Okay, so these 5 points are the reasons why we have picked up bioreactors, and even we can culture large amount of substrate in it. This is one reason. And bioprocess engineering, which was one of the bio principle of the biotechnology chapter, was fulfilled using the bioreactor. So all these points can be included in your answer. Next question is, following is the sequence of nucleotide in 2 strands of DNA. Observe the strands. They are given the strands here. Now, name the spatial term used for such an arrangement of the nucleotides. Now, if you look into the nucleotides, you have moved here, it is GART-TC, and here you move on, it is GART-TC. So such type of mirror images, they have got a special term, and which is known as palindromes. So answer to this will be palindrome. Name the spatial type of enzymes which function at these specific points. So these special enzymes are restriction endonucleases, which act on the inner side of the DNA. Next is, name the enzyme that cut DNA between GA sequence. Here they are asking which exactly is the restriction endonuclease which acts between GNA. So it is ECO R1. Okay. Now next question is, from what you have learned, can you tell whether enzymes are bigger or DNA is bigger in molecular size? How did you know? Now if you compare enzymes with DNA, of course DNA is a bigger biomolecule. Why DNA is a bigger molecule? Because we know that DNA can self-replicate, DNA can transcribe to from RNA, RNA can translate to from the proteins, and these proteins may function as enzymes. So enzymes are derived from DNA. If DNA will not be there, enzymes will not be there. So of course DNA is a bigger molecule, and you need to explain the concept of central dogma in how did you know. Okay, this is one of the good questions. Next is, mention the number of primers required in each cycle of PCR and write the roles of primer and DNA polymerase in PCR. Remember, the steps of PCR were denaturation, then annealing, and then it was extension. In denaturation, the 2 strands of DNA that have to be amplified, they will move apart from each other. In annealing process, basically what happened, primers were required, right? So primers, they used to adhere at the 5' end here, and here they would be this side. So if we have taken 1 double helical DNA, how many primers we have seen here? 2 primers, right? So here the answer will be, mention the number of primers, it will be 2, and write the role of primer. So primer is a short stretch of DNA, approximately 8 to 12 nucleotides long, and DNA polymerase cannot initiate the process. It can just help in elongation, which we have read in chapter number 6, right? So we'll be writing that primers are the ones that helps in initiation of the DNA replication, and DNA polymerase is the one that only does the elongation, right? So role of DNA polymerase comes in the extension process. This also has to be written. DNA polymerase does its role in the extension step of the PCR, whereas the primers, they play an important role in the annealing process, right? I hope this is clear. Now we move on to next one. What are bacteriophage vectors? So whenever we use bacteriophage for transferring the recombinant DNA, or whenever we want to use the gene of interest by using bacteriophage as a vector, they become bacteriophage vectors. And very important bacteriophages which are used as vectors are M13 and phage lambda. Okay, phage lambda and M13, they are being used as bacteriophage vectors. You know, bacteriophage vectors are more helpful because whenever we want to have gene of interest in a larger quantity, means if the DNA has larger number of base pairs, then at that time we are being suggested to use the bacteriophage vectors. Next question is, a recombinant DNA is formed when sticky end of vector DNA and foreign DNA are joined. Explain how sticky ends are being formed and how do they get joined. Now for this question, this is purely a theoretical question. For this, I will be sharing my video in the I button. You can go through it and it will help you in answering this complete question, question number 14. Okay, you'll get the link of it on the top right side. Name, question number 15, name the organism in which vector shown is inserted to get the copies of the desired gene. So if you will look in it, they are talking about PBR322. So we have read it in CRT that they use the organism E. coli for this purpose. Mention the area labeled in the vector responsible for controlling the copy number of the inserted gene. So here the area which is responsible is the ORI site. ORI site is the one that has the origin of replication. So it also decides that how many copies of an organism could be formed, right? So answer to this will be the ORI site. And you can show with an arrow also like this. 15 part 3, name and explain the role of selectable marker in the vector shown. Now here the selectable marker is ampicillin resistant gene and tetracycline resistant gene. For this also I will be sharing a button in the I link. Just go through that video because that will explain you what is plating and when and why plating should be done, what is the other way out. Okay, so for this also just click on the video in the I button. I hope you have understood all the questions. We have some more questions from the same chapter. Stay connected and do not forget to subscribe to my channel. Happy learning.